Good morning, everyone. I am Dr. K. Subhas Shankari. Today, we are going to learn about plant tissue culture. So, what is plant tissue culture? As we all know, plant and tissue culture techniques, isn't it? So, how we are going to do that? Okay. So, what is plant tissue culture? How to culture in vitro condition? These things we are going to learn in this chapter. Okay. So plant tissue culture is just nothing but it's a technique of growing plant cells, tissues, organs, seeds or other plant pods in a sterile environment on a nutrient medium in vitro condition. Inside the lab condition with the required nutrients, minerals, hormones, we are going to grow a plant cell, tissue, organ, seeds or any other parts we can grow in a optimum culture conditions in an in vitro. Okay, and why we do this plant tissue culture? Any idea? Before we are going to do this, we should know the importance of the specific plant. Okay, then only we should know about. Okay, it may be either the leaf having some medicinal properties or the entire plant also having some medicinal properties. So in such condition only we are going to do not only for medicinal, some other economical and um, any other good flowering plants, good fruits. They are having good fruits and some desirable traits. These are all maybe some reasons of doing tissue culture techniques. Okay, so in this process, we can quickly produce a mature plant. The production of multiples of plants in absence of seeds or necessary pollinators to produce seeds. Okay, we can produce seed also. In this techniques, we can produce not only for a mature plants, we can produce the seeds also. The regeneration of whole plants from plant cells that have been genetically modified. Okay, we can use genetically modified. Not only we can use the whole plant which is similar to the parent plant, we can also modify the plant using genetic transformation methods. So here we can see that purple color cauliflower. I think you may can see. So how it possible through this technique only it made possible. Okay, through this genetical transformation techniques we can do this purple color cauliflower. Okay, and also the production of plant in sterile containers reduces disease transmissions. Yes, of course. If we are using these plant tissue culture and this in vitro techniques, our plant couldn't, uh, what we can say, face any stress related or disease related issues. So, this it could be a good thing, no? And also, it allows the production of plants from seeds that otherwise have very low chances of germinating and growing. For example, orchids and nepenthes. To clean particular plants of viral and other infections, to quickly multiply these plants as a cleaned stock for horticulture and agriculture. So we can minimize the infectious rate during cultivation period. In this, if you are doing for if you are go for a plant tissue culture, we can also minimize the infectious ratios. Okay, and what are the terminologies we should know in this plant tissue culture techniques? Okay, the first one is explant. What is the explant? Explant, which is nothing but the living tissue which is transferred from a plant to an artificial medium for culture. After that, it can be any portion of shoots, leaves, roots, flower, or cells from a plant, which means you just consider this diagram which denotes a whole plant and the nearby one tube is there. So, and also some arrow mark which is denotes stem, apical tissue, leaves, roots. What it actually means, what is the connection between the explant and this picture? You just think it off, okay? See, the living tissue means it can be either the apical, the tissue from apical or leaf or nodals, shoots or roots. It can be either, it can be any of the plant pods, can also transfer in, if it is transferred to the artificial medium. The tube one is the tube is having some green colored substrate is, we can see, if it is transferred to the artificial medium, that is nothing but our ms medium, that portion it can be transformed into shoot, leaves, roots, flowers, or cell. Any part of the cell it can be differentiate. So what is the expand? The first one, what we are going to choose. Okay. If I choose our leaf portion, I am going to inoculate the leaf portion into the MS medium means that is my explant. If I going to cut my shoot and which is transferred to my MS medium means that is my explant. So what, which of the plant pot which I am going to choose, which one I want to transfer to a whole plant, that is our explant. Okay, and how it has happened. The previous slide that is clearly explained what is explant. Explant, it can be any part of the plant pot. Okay, explant is any of the plant part can consider as a explant. Okay, how it is possible? The any plant, any part of the plant, if I put a leaf means it might be a grow as a leaf. How it can be grow as a whole plant? Do you want to? Do you want to know the answer? See, the answer is totipotency. The adult plant cells are totipotent. Okay, adult plant having the totipotent cells. Okay, meaningly they have the ability to give rise to a fully differentiated plant. Because of this, it is possible to collect cells from a mature plant and use those cells to produce clones of that plant. That means, in previous slide, I talk about the explant. Each of the plant pod which is having the totipotency. Totipotency means the cells it can able to grow a fully differentiated plant. Okay, if I put a root into artificial medium, it can be able to grow a complete matured plant, which means that root cells having the totipotency. No, this totipotency which leads to gives rise to a whole mature plant. So in this diagram, what you understand, this diagram, the root of carrot plant which is inoculated into the MS medium, and the single cell which is cell division takes place here in this culture media, cell division takes place. It will propagate from single cell to multi-cell, and then the defined callus is and next introduced into the media, which gives rise to the adult plant because of the totipotency. Yes. Yes. Plant tissue culture basics. Modern plant tissue culture is performed under aseptic condition. What is the aseptic condition? It's a completely pure conditions. Okay. Living plant materials from environment are naturally contaminated on their surface. Yes, it is, of course. And sometimes interiors with micro, not only for environmental source, with some microorganisms. So, surface sterilization of starting material, that is our explant. It may be either a shoot, root, anything else. Okay, leaf, anything else. So the starting material should be surface sterilized. Okay, in chemical solution, usually alcohol and sodium or calcium hypochlorite is used in a lab. Okay, so first, what thing we should know in plant tissue culture? Everything should coming under aseptic condition. The second thing is the surface of the environment should be cleaned, and the explant should be undergone surface sterilization using alcohol and sodium calcium hypochlorites. Okay, the next one, in plant tissue culture basics, we should know the explants are then usually placed on the surface of the solid culture medium. Okay, after surface sterilization, our explant should go to the solid culture medium, but are sometimes placed directly into the liquid medium when cell suspension cultures are desired. In such conditions, if some explants need a suspension medium, that time we should recommend a suspension culture, or else we directly go for the solid medium. The next one is a culture medium. Culture media are generally composed of inorganic salts plus few organic nutrients. Inorganic salts means are minerals. Later on, we should know. And few organic nutrients, vitamins, and plant hormones. Okay, plant tissue culture media. What it actually composed? First, salt mixtures, organic substances, natural complexes, inert supportive materials, and growth regulators. We'll, dis- we'll see in detail. These are the macronutrient salts. See the macronutrient salts: ammonium nitrate, potassium nitrate, calcium chloride, magnesium sulfide, potassium hypophosphate, iron or sodium EDTA, boric acid, manganese sulfate, zinc sulfate, potassium iodide, sodium molybdate, cupric sulfate, and cobalt sulfite. And macronutrient salts: nitrogen, phosphorus, potassium. See how many macronutrients which will help to grow a whole plant. Nitrogen, which is influences the plant growth rate, which is essential in plant nucleic acids. Okay. Wait a minute. Nitrogen, which is essential for the nucleic acids. Proteins, not only that, proteins, chlorophyll, amino acids, and hormones. And phosphorus, which is abundant in meristematic and fast growing tissues, which is highly present in the meristematic and fast growing tissues, essential in photosynthesis respiration. And potassium, which is necessary for cell division. Meristematic tissue helps in pathways for carbohydrate, protein, and chlorophyll synthesis. Why we give prominent focus on these three macronutrients? Yeah, in small class we learned this. NPK, yes. These are all the major macronutrients, just having the playing the most prominent role in plant growth promotion. Okay, and also calcium, iron, magnesium, and sulfur, manganese. These are all the part of it. Okay, and when coming to micronutrient salts, molybdenum, zinc, boron, copper. These are all the most important micronutrient salts which is important for the plant growth promotion. Okay, and these are all the applications. Right. The other organics we are used is organic acids, phenolic compounds, and these are all the natural complexes: coconut endosperm, fish emulsion, protein hydrolysates, tomato juice, yeast extracts. See, see, this is the flow diagram of the procedure of doing surface sterilization. First, they are choosing the explant and wash with the water, and then again wash with the tap water. Then the surface sterilization is done by slicing the explant using the sterile scalpel, using ethanol and sodium calcium hypochloride. And after cutting and sterilized environment, the explant will insert into the MS medium. Okay, so each and every technique should be in aseptic condition. Every part of the technique should having the optimum time period. If it excess or less, it will affect the callus growth. Definitely, it will affect. So each and every step should be careful. Even it might be incineration or handling techniques. Everything should be a clear and thorough knowledge. Okay. So in this class, what we see, what is explained, what about the totipotency, what is surface sterilization, what are the basics in plant tissue culture. Okay. And these are all some applications of doing plant tissue cultures. So why we need to go for plant tissue culture? In the early part of this chapter, I'll denote that to conserve the rare and endangered plant species. Okay, we need to grow the large scale growth of plant cells in liquid cell culture bioreactors for production of valuable compounds like plant derived secondary metabolites and recombinant proteins used biopharmaceuticals. See, we need to use a commercial values of plants. Plants having some commercial purpose. We are using some secondary metabolites and enzymes. There's lots of n number of vast variety of properties the plants having. So in that reason, uh, we couldn't exploit uh, we couldn't exploit the plant. Okay, In that concern, we are doing this culturing techniques through the recombinant and transformation techniques. We are doing this productions in commercial scale. Okay. The next one is rapidly study the molecular basis of physiological, biochemical, and reproductive mechanisms in plants. For example, in vitro selection of stress tolerant plants. Why we choose this stress tolerant plants? See, if your stress tolerant plants having the potential to grow in a drought and stress resistance. So in that kind of plant is basically having the capability of immunoprotective disease control genes in nature. So we should know that molecular basis of physiological, biochemical, and reproductive mechanisms of these plants. Okay. And the sixth one is to cross pollinate distantly related species and then tissue culture the resulting embryo which would otherwise normally die for embryo rescue. Okay. For that purpose, also, we should go for this culturing techniques. Okay. As a tissue for transformation, followed by either short term testing of genetic constructs or regeneration of transgenic plants. Yes, we should do the regeneration of transgenic plants. Okay. Certain techniques such as meristem tip culture can be used to produce clean plant material from Virtued stock such as sugar cane, potatoes, many species of soft fruit production, identical sterile hybrid species can obtain. Yeah, in some marketing aspects, the fruit ripening is a some issues in marketing industries. So, in that condition, they are using the hybrid technologies, GMO tomatoes, potatoes, brinjals. There are a number of GMO fruits and vegetables are available in markets. They are having some nutritional values as well as ripening control skins. Okay, and large scale, the final one is a large scale production of artificial seeds through somatic embryogenesis. Somatic embryogenesis, which is we already know somatic embryogenesis, which is they don't have that embryo, it doesn't originates from the zygotes, okay, gametes, which is from the somatic cells. So that large scale production of artificial seeds in using these somatic embryogenesis, they are using some artificial seed productions. Okay, in that concern also we are doing this tissue culture techniques. Okay. Okay, thank you, girls. And next slide, next class, we will deeply discuss about the culture techniques of callus, androgenesis, and haploid cultures, embryogenesis. Lots of culturing techniques we'll discuss in the next class. Okay, thank you.